I think you have gone through books and have acquired knowledge regarding this type of figure, action for monophasic action potential. And here is the stimulating electrode, cathodic stimulus, that is responsible for the depolarization and ultimately the generation of action potential. And these are the different, and this figure composite diagram, and it contains the action potential. Action potential. Initial part of the action potential up to this level is the electrotonic depolarization, and it is the firing point corresponds to minus 55 millivolt. Whenever there is abrupt change in the membrane potential, it is the action potential. And here the recorder, and in which one electrode is on the outside and another inserted into the membrane. So the resting membrane potential here is stable potential, and its magnitude is minus 70 millivolt. That is inside in comparison to outside. It is expressed in this way. Now, whenever there is the stimulus and it is above the threshold stimulus, then there is generation of action potential and it will propagate. And whatever may be the effort of, there is the blocking, and there is some leaking of charges at the beginning of the, time of stimulation. Whatever may be the effect, to obscure it, there is some change in the recording at this time of stimulus. So it is the point of stimulus, and it is the beginning of the action potential. And so this part is the latent period. Latent period. Yeah. And from the latent period and this distance, you can find out the conduction velocity of the action. And this type of experiments were done in invertebrate actions, giant actions of squid or so, where the diameter is such that this type of experiment can be done. And behavior of the mammalian actions, whose diameter, action diameter less than 20 micrometer or so, and that is difficult to do this. But in this case, the behavior is the same. So this type of experiment has been done. And the different parts you can see, that is the initial part of the action potential. It is relatively slow, and it is the local potential, local response. Initial is a pure electronic response, cat electrotonic response. Then there is some additional response due to gradual opening of the voltage gated sodium channels, and that is called local response. And when it reaches this value of minus 55 millivolt, then there is abrupt opening of the good number of voltage gated sodium channels, and that overwhelms every attempt to bring it back to the normal level. And then there is runaway depolarization, and up to a level of plus 30 millivolt. And it is due to the increase in the conductivity of the sodium ion. And it were initially it is known that it is the sodium ion is responsible And there are some experiments done, voltage clamp technique, by means of which a particular potential can be introduced by a monitor, by a stimulus, and it is held in that fashion. And in this way, sodium current, potassium current, and by blockers or eliminating sodium ion, in this way different experiments have been done. In the older texts, they described this, but current text, physiology text, they ignored it because it has been known, so they ignored it, every attempts. But older text, they explain in very good way how it was known, that it is the sodium ion, entry of the sodium ion is responsible for this, and it is called GNA, conductance. It is a measure of permeability. It is reciprocal of the resistance, electrical resistance. So it is a measure of the permeability, conductance, and it is expressed in millimole by centimeter square. Per centimeter square, millimole in this way. Suppose it is 30 or so in this way, and it is the potassium, and this is the potassium. It is the potassium GK. Potassium. And it's a different part, that is the abrupt rise and rapid fall, is called the spike potential. Spike potential. So you can spike. And this part, that is the above the zero level, if you consider, then it is the overshoot. It is called overshoot. Clear? Overshoot. And then there is the repolarization. From this part to this part is the depolarization, and this remaining part is repolarization. The later part of the repolarization is relatively slower process. After 70% or so, and it is called after depolarization. And then it crosses the resting membrane potential line to hyperpolarizing direction, and it is called after hyperpolarization. This is a quick process occurring in milliseconds. So what occurs during this action potential generation? There are in the membrane, there are voltage gated sodium channel in the cell membrane of skeletal muscle, and the sarcolemma, plasma membrane of skeletal muscle, or even in cardiac muscle, there are voltage gated sodium channels, and that open abruptly in huge numbers, so that there is reversal of the potential at a particular point. So inside become positive. Yeah. And then repolarization starts, and it is due to exit of the potassium ion. Sodium channels open quickly, and also rapidly it becomes in the inactivated state. It is a one form of inactivation. There is abrupt opening. There is abrupt opening of the sodium channels, and after just opening, within a short period it becomes inactivated. It is closed. There are two gates, outer gate and inner gate. They express it, textbook express in this way. Whenever there is entry, both the gates are open, but then the inner gate, they become closed, and it occurs quickly, rapidly, and it becomes inactivated, and it can be reactivated when it comes to the normal position. Normal position. And it is due to the behavior of the sodium channels that is formed of 4 subunits, and they have some component that behaves in this way. Quick opening, and after a short period, within milliseconds, it becomes inactivated and it remains for some state, and then when it acquires the original state, normal state, then it can be activated again. Inactivated state. So, before the end of this spike, after the end of the spike potential, then its conductivity remains 0. Abrupt increase and abrupt decrease, it is the GNA. Yeah. Conductance, reciprocal of resistance. Resistance unit is ohm and opposite is MHO. And M milli MHO oblique centimeter square is this, in this textbook they given in this way. Yeah. Now potassium channels begins to open, voltage gated potassium channels begin to open, but it is a slow process and it is not peak value is occurs sometime less after this. It is, and it also closes slowly, and so, and it is responsible for the after hyperpolarization. Yeah. Potassium channel. Now it is asked that whether, why, what value it should attain. Equilibrium potential of sodium ion you have read in the text, and it is plus 60 millivolt. Plus 60 millivolt. And if the sodium channel is only responsible, it should try to attain this value, but it falls short of that value. Plus 60 millivolt is never attained. Clear? What is the reason of this shortfall from the plus 60 millivolt? There is this closure of the sodium channel is opening and closure is the, both are rapid process. One thing. Second is that whenever there is this overshoot, then this there is the electrical gradient in the opposite direction. Normally resting membrane potential is minus 70 millivolt, and sodium concentration is higher in the much higher in the extracellular fluid than in the intracellular fluid. You have to know these values. And also the Nernst equation of sodium ion. Yeah. And plus 60 millivolt it should be, but it does not reach that value. And the third cause is that at the same time the voltage gated potassium channels they begin to open. It is a relatively slow process, so that there is the exit of, there is the entry of the sodium ion, entry of the counteracted by this potassium ion, so that ultimately that value is not reached. Bujha gelo? And then this potassium is coming out and exit of the potassium ion is primarily responsible for the repolarization, because the sodium channels are closed, and repolarization. The EK, equilibrium potential of potassium, is minus 90 millivolt. Minus 90 millivolt. You know this formula. I have written this formula in the book, and I think you have gone through the books, and they have given the formula. From natural logarithm it is brought to base 10 logarithm, and at a particular temperature in a different text they have given a ready-made formula, so that you can understand them easily. Remember them. And in case of potassium ion, it is minus 90 millivolt. But the resting membrane potential is minus 70 millivolt. So there is more potassium when you consider the case of potassium alone. And it will try to membrane potential to its equilibrium value of minus 90 millivolt. But the existing value is minus 70 millivolt. So you can imagine that there is some extra potassium ion inside, and it will try to go to the outside to maintain the equilibrium potential. According to the concentration difference that is maintained by this membrane, sodium via the sodium potassium ATPase that maintains this value. Clear? And so after depolarization is a relatively short time, but it can be prolonged. And it should be understood that whenever there is change in the membrane potential, action potential inside become positive due to entry of the sodium ion. But this ion increase in the ion of sodium is minute fraction of this present. Separation of charges is not an easy thing. The membrane behaves as a capacitor, and it is not easy to maintain this charge difference. Clear? Number of ions in comparison to these ions everywhere, it is a negligible fraction. In spite of that, it can be demonstrated. By repeated stimulation, it can be demonstrated. And it can be calculated that what is the delivery of the charge to maintain this capacitance of the membrane. Excitable tissue, nerve or muscle, in this case, this average capacitance is 1 microfarad per centimeter square, and it is maintained. But the number of ions are negligible in comparison to the others, and only at the level of the membrane, nanometer, their dimension, nanometer, and only at that site there is separation of charges. You have read capacitance in your higher secondary course, and the first MBBS course is nothing but some continuation of the higher secondary course. Yeah. So this subject is extremely easy, you think, and you can go through books and have adequate knowledge of this. Yeah. Microfarad, 1 microfarad per centimeter square is the capacitance, and you know the formula. Q is equal to C into V. Q is that is the total electricity in coulombs, and it is the capacitance multiplied by the voltage. And in this way it can be calculated, and calculation is given in older texts. I cannot remember everything in detail, but they have calculated, and they have shown that their concentration in comparison to this total amount of ions present elsewhere, ICF, they are negligible. In spite of that, there is the generation of this potential, and this potential is required for the development of the action potential. What is the development? What is the requirement? That is the action potential. It is responsible for the action of the excitable tissues. Suppose whenever I am speaking, everyone is speaking, then action potential is coming from the neurons from the medulla or brainstem into the facial muscles, and it is the action potential that is present. Action potential, and this action potential is responsible for the secretion of neurotransmitter, and by the neurotransmitter, ultimately there is action potential generation in the skeletal muscle membrane, and it will spread, and in response to this action potential it can contract. Action potential generation is the only function of the nervous system, and in response to this there is contraction of muscle, secretion of glands, and many other functions done. Clear? Now it will propagate, and you have also learned the all or none law. All or none law you have read in this textbook. whenever it reaches the threshold, if the other circumstantial conditions remain same, the shape size of the action potential is unaltered and it will propagate with the same silhouette. মানে ওর মতো যে চেহারা ওইটা দিয়ে সে করবে, there is no change. If you increase the stimulus, supramaximal stimulus, and there is no change. When it is below the threshold level, there is no action potential generation. Below the threshold, subthreshold stimulus is responsible for what type of potential in a neuron? কি ধরনের পোটেনশিয়ালের উৎপত্তি হয়? ইলেকট্রনিক পোটেনশিয়াল। এটা একজন না, সবার এটা জানতে হবে, এর মধ্যে কিছু নেই, সহজ জিনিস। ইলেকট্রনিক পোটেনশিয়াল, লোকাল রেসপন্স। হোয়াই ইট ইজ কল্ড ইলেকট্রনিক অর লোকাল রেসপন্স? কি জন্য এটাকে বলা হয়? যে ইলেকট্রনিক পোটেনশিয়াল বলছে সে তোমার বইয়ে কি বলছে? এটাকে কেন লোকাল রেসপন্স বলা হয়? হ্যাঁ হ্যাঁ, ট্রাভেল করতে পারে না। তার মানে আমি যদি 1 মিলিমিটার 2 মিলিমিটার দূরে রাখি তাহলে আমি তার রেসপন্সটা রেকর্ড করতে পারবো, কিন্তু আমি যদি 1 সেন্টিমিটার দূরে রাখি তাহলে তার কোনো রেসপন্সই সেখানে বোঝা যাবে না, তার মানে সেটা প্রপাগেট করবে না। কিন্তু তোমার যদি অ্যাকশন, ইফ দেয়ার ইজ অ্যাকশন পোটেনশিয়াল জেনারেশন, it can distance up to 1 meter or so, 1 meter or so, because the length of the action supplying the muscles of the foot, that we can move the toes voluntarily, and it can travel up to 1 meter from the spinal cord. Anterior horn of the spinal cord, the distance is 1 meter or even more than that. And if there is action potential generation, it will automatically move this. Yeah. But there is, whenever it is sub-threshold, there is a local response, but it cannot propagate. So now that we are in the position to know what occurs, but by which means it can spread at first in the unmyelinated neuron, and the same principle is applied to myelinated neuron. In unmyelinated neuron, it is present in the autonomic nervous system. Post-ganglionic in the motor side, post-ganglionic, it is the unmyelinated neuron. There is no myelination. Myelination does not mean that it has no cellular coating. There is cellular coating, but there is no multiplication of its plasma membrane. It is the basic histological findings. Suppose it is the action 1 μm in diameter. In case of unmyelinated action, there is a Schwann cell coat. Single, there may be in this way and they come close together, but there is no rounding of this. And in case of myelinated neuron, suppose it is, commonly it is more than this 5 μm or so, and in that case there is folding of this. Folding of this Schwann cells. Nucleus. And it is the nucleus. Unmyelinated does not, it means that it has no coating, it has single coating, and that gives some protection and some other functions. But in case of myelination, there is a rounding and wrappings. ঘুরে ঘুরে আসবে এরকম ভাবে, বুঝতে পারছেন? সাপের মতো জড়িয়ে জড়িয়ে ধরবে, ধরে না সেরকম দেখবে। অজগর সাপ যখন একটা মানুষকে মারবে তখন কি করে, সে জড়িয়ে ধরবে, ধরে চাপ দেবে এইভাবে সেইভাবে, তাই সেখানে এরকমভাবে র্যাপিংস হবে, র্যাপিংস, দ্যাট ইজ এ myelinated. A single cell is wrapping. In case of peripheral nervous system, a single cell. In case of unmyelinated action, if you consider the bare action. You consider the bare action, and then there is a stimulus is given. A stimulus is given. It is supra-threshold. And in case of stimulus, you know that we are considering here this rectangular type of impulses, and it is the magnitude, it is the strength, and it is the duration. Suppose these are the sub-threshold value, and it is the threshold value, and these are all supra-threshold value. So when it is threshold value and above, then there is some change in the membrane potential. Normally, everywhere on this side and on this side they are all positive. But whenever there is the stimulus is given, and in that case there is reversal of the potential. Inside become positive, outside negative. And whenever there is action potential generation, its magnitude is much more, at least to minus 55 millivolt it will reach at that point. And what occurs? And it will, then there is electronic charge generation initially. So it will flow in this way. Positive to negative is our convention. Positive to negative in this convention. And in this way, neighboring part, they becomes depolarized. Initially there is electronic depolarization, but when ultimately it reaches, whenever there is action potential generation in one spot, then there is action potential is developed in this way. Initially there is action potential, and at that place then there is action potential. It will propagate in this way, and in this way it will also propagate. Action potential. And when this part is becomes this, and then the same thing will happen, and it will spread, and in this way another action potential done, neighboring one, and in this way circular current will flow, and it will spread in this way. So from spot or on either direction it can move. When action is cut and brought in the laboratory, and in that case you are giving stimulus to any middle point, and from that point onwards then there is circular current flow. The circular current flow initially causes depolarization, that is electronic depolarization and local response, and ultimately it will reach to the firing level, and it will spread, and in this way they will spread in this fashion. So it is the active participation of the membrane. Clear? Conduction velocity, and in this way it is conducted in this way. In both sides it can conduct in the laboratory preparations, but normally within the body it will conduct in a particular direction according to the requirement of the system. Circular current flow, neighboring, and every part of this neuron is utilized in this case. It is reacting to this, and it will propagate in this way, circular current. And so you can compare it with that of the train of a gunpowder. যেরকম তোমার ওই তারাবাতি যেমন জ্বালায়, তুমি একটা দিকে তুমি যখন ডিস্টাল পার্টটা তুমি যে আগুন লাগলো দেখবে it will ignite the neighboring particle. The neighboring particle will then ignite this another particle more proximal. In this way it will propagate. And when it starts, it will go to the end, if not blocked. It is the same process. But whenever there is a subthreshold stimulus, then it will not propagate. Cathedral stimulus is the stimulating electrode because it is depolarizing. And in this way it will spread. But it is a relatively slow process. Yeah. And fast process is required in some cases. In invertebrate axons, there is small creatures, and so this is sufficient for them. Nature they have highest intelligence over every creation, and their creation is unique and complete. And according to the requirement of the animal, and in that case, there is no need of much higher velocity of conduction. So that is sufficient. Change in the diameter is responsible. A squid action of 500 μm diameter, and it has conduction velocity of 25 centimeter per second. But this type of, it is adequate for that small animal, but like human beings, it is not adequate. Reflex responses are not adequate for human being. Yeah. With this speed, then it is difficult to protect from the body part from injury. Suppose if I have a prick in the toe, it will come to the spinal cord, and the removal of the toe will get 2 meter distance. 2 meter distance. If 25 centimeter is the rate, so you can imagine what is the time required. Apparent different, and within this long period there is process will continue, but we have to turn it quickly, and for this reason there is the development of the process by the nature that is called myelination, myelinated. Myelination is done with the help of the wrappings of this membrane, Schwann cell membrane in peripheral nervous system. Nervous system. And in case of myelinated nerve, there is this saltatory process. There are nodes of Ranvier. In that case, what is occurring in that? In case of myelinated neuron, there are myelin sheaths. 1 to 2 millimeter distances, myelination is there. Myelin sheaths. These are the myelin sheaths. So whenever there is the action potential is progressing, suppose you can take the case there is this the action potential is progressing, and what occur? Then it is the insulator. It is the insulator, and it is the cable properties of the neurons or muscles. Cable properties. You will given in the textbook CABLE, cable properties, and everywhere you see that there is a coating, there is the insulator coating, and is that there is the electricity is not lost through it, and there is insulating material in which this resistance is very high, and almost there is nothing of electric energy can come out of the core of that. Yeah. So there is no loss of energy, electrical energy, and it will affect only the node of Ranvier, and the intermediate this space 1 μm or so in their dimension, 1 μm, and this distance is 1 to 2 millimeter. and then it is the nodes of Ranvier, VIER, nodes of Ranvier. And whenever there is action potential generation, then it will the same electrotonic or circular current is also flowing. Circular current is also flowing, but it involves in not this part. It is involving this node of Ranvier, and their distribution of this voltage gated sodium channels is highest in the node of Ranvier. So very quickly it will, if there is action potential at this point, and at this point very quickly the action potential, and it will influence in this way, and in this way it will propagate quickly. Clear? 500 μm diameter, 500 μm diameter action, unmyelinated action conduction velocity of 25 meter per second. And our actions of the motor neuron or some sensory neurons, A alpha, 10 to 20 μm diameter, and its conduction velocity is 70 to 120 meter per second. So you compare. In case of unmyelinated actions, every part of the surface of this membrane is to be involved, and it is a much slower process. In unmyelinated neuron, that is in case of the action over which this type of experiment has been done, and in that case it is only 25 meter per second. But in case of A alpha neurons, that is the motor and proprioceptions, and they have a thickly myelinated, and their conduction velocity and their diameter is, axon diameter is 10 to 20, and this conduction velocity is this. In case of 5 μm myelinated action, and its conduction velocity is approximately 30 meter per second. There are classification of these neurons. So it is the mechanism by means of which it is taking place. And there are the terms, that is the length constant and time constant. Length constant and time constant in this way. Whenever there is insulation, there is this fall in the capacitance. Simply it is the Rm it is called. It is called this Rc, time constant. Resistance multiplied by this capacitance. Yeah. Capacitance whenever there is, so whenever the time constant is low, their conduction velocity will increase. Similarly, when this resistance in comparison to the inner resistance increases, this conduction velocity increases. Yeah. If you consider this unmyelinated action, this membrane is the insulator, and it has a capacitance because it is separating charges, capacitors. And within this, in the cytosol, there is the electrolyte fluid, and there through which the electricity can propagate freely. So their resistance is lower. So ultimately what results is this membrane resistance and this resistance in this direction, Rm by Ri, and it is the equal to length constant. Roughly elaborate given in test of older editions of Barnley or so, that you can consult. But this time constant is this value and this resistance and capacitance circuit. And whenever there is rounding, then what occurs? There is the fall in the capacitance and increase in the resistance. Capacitance, when they are in series and their reciprocals are added. Reciprocals are added. Yeah. And in case of resistance, when they are in series, they are directly added in the circuit, electrical circuit. Suppose if there is 25 this type of fold, then there is the membrane, number of membrane is 50. So the total capacitance, suppose the CT is equal to 1 by C plus 1 by C up to 50 times. So is it 50 by C. So CT is equal to C by 50. But in this case of resistance, it is simply added when they are in series. So the length constant increases, length constant Ri remains the same. So there is the time constant is reduced and the propagation velocity increases. It is applied equally well in unmyelinated and in non-propagated local response, electronic response, and also in the action potential you can, this concept is applicable. And it is the nature's device. It is in nature's device, so that with this small space it is required. Yeah. Otherwise, the basic feature, এই যে একটা মানুষের বা অন্যান্য প্রাণীর যে একটা সুন্দর যে পিকচার, এই একটা সুন্দর একটা পিকচার আছে না, প্রত্যেকের মানুষের একটা সৌন্দর্য আছে, সেটা থাকতো না, কদাকার হয়ে যেত। এখানকার যে নার্ভের সাপ্লাই হতো, প্রত্যেকের নার্ভের ভেলোসিটি যদি ওরকম হতো, ওই পরিমাণ নার্ভ, একটা হিপোপটামাসের মতন চেহারা হতো মানুষের, সেটা হয় না। নেচার যে সৃষ্টি করেছে, সে তার সবচেয়ে বুদ্ধিমান। সে যেখানে যেখানে দরকার, যতটুকু ফিজিক্সের দরকার, যতটুকু ম্যাথামেটিক্সের দরকার, সে ততটুকু নিখুঁতভাবে করে গেছে। এবং এটা বুঝতেই আমাদের হিমশিম অবস্থা হয়ে যাচ্ছে। বোঝা গেল? কাজেই তোমাদের আমার বলা রইলো, তোমরা সবাই বুদ্ধিমান আমার থেকে, এটা একদম আমি সার্টিফিকেট দিয়ে দিলাম। তোমরা প্রত্যেকে এগুলো পুরনো এডিশন থেকে ভালো করে দেখে নেবে। আমি সার্টিফিকেট দিয়ে দেবো তোমাদের যে আমার থেকে বুদ্ধিমান লোকেরা যাচ্ছে, এদেরকে হেল্প করুন। এরা আরো গভীরে যেতে চায়, ল্যাবের দিকে সেটা দেখাবে, তখন দেখালে পরে তোমাকে সেরকম বই বার করে দেবে, তুমি সেটা পড়বে এবং পড়লে শর্ট করে বুঝতে পারবে। বুঝতে পারছো? যাই হোক, and this is this behavior of this, so that there is the time constant, mean time that is this within which time it becomes stimulated, and it is occurs exponentially. We discussed other day, but today there are some other topics that is also important for the examination. Refractory period. It is given. This type of complicated things will not given to you, because you have come here to acquire some knowledge, so that can be applied in medical sciences for the treatment. Details, this type of physics, mathematics is not required for you. For this reason, the newer text, they shorten this part, inversion, insulation and saltatory conduction. It may be come in the examination, but the knowledge has no bounds. Like the greed of the human beings, they have no bounds. Similarly, knowledge has no bounds. And so it is our duty to create some interest amongst you that much more things can be known, can be done, can be, and then the conception will be perfect for the medical science particularly. But in general, it is not much required for your clinical concept, for the treatment of the patients. If you know how to go through these nerves, go through these blood vessels and given a drip, it is sufficient for the patient. জিনিসটা বুঝতে পারছো? একটা লোক কলেরা নিয়ে এসছে বা গ্যাস্ট্রাইটিস নিয়ে এসছে, ফ্লুইড দেওয়ার দরকার, তখন তোমাকে কেউ অসমোলারিটি তোমার চিন্তা করলে হবে না, তোমাকে জানতে হবে কি করে ব্লাড ভেসেলটাকে পিয়ার্স করে একটা ক্যাথিটার ঢোকাতে হবে, যেই ক্যাথিটারটা ওই সোডিয়াম ক্লোরাইড সলিউশনের সঙ্গে যুক্ত আছে এবং সেটা গেলে পরেই তোমার রোগীর উন্নতি হবে। কাজে সূক্ষ্ম জ্ঞান না হলেও সেখানে কাজ চলে যাচ্ছে। এর জন্য দেখা যাচ্ছে অনেক হেলথ সেন্টারে কেউ নেই, কিন্তু যারা ডিউটি করছে গ্রুপ ডি এমপ্লয়ি, তারা ওটা জেনে গেছে, তারা ওটা দেখে গেছে যে ডাক্তারদের সঙ্গে বা সিস্টারদের সঙ্গে কাজ করে যে এটা কিভাবে করতে হয়, এবং তারা অনেক রোগীর প্রাণ বাঁচাচ্ছে। ওয়ার্কিং নলেজটা এখানে কম মূল্যবান মানুষের জীবনের ক্ষেত্রে, এবং দেখবেন লোকেরা তাকে ডাক্তারবাবু বলছে এবং সে কাজও করে যেতে পারছে। কাজে সব জিনিসেরই সূক্ষ্ম জিনিসের জানা দরকার, কিন্তু সেটা সব ক্ষেত্রে অনেক সময় প্রয়োগের প্রয়োজন পড়ে না বা মানুষের সম্ভাবনা হয় না। কিন্তু যখন এই সামান্য কয়েকটা মাস আমাদের কাছে কন্ট্যাক্টে আসছো, তখন আমাদের উচিত যে অন্তত পয়েন্টটা ধরিয়ে দেওয়া, যাতে ইফ ইউ ইন্টারেস্টেড ইউ ক্যান প্রসিড and go into the depth. Now refractory period is asked in the examination. Absolute refractory period and relative refractory period. Refractory period, that is, suppose up to 1/3 or roughly 1/3 of this action potential to the firing level, if you consider, then it is the absolute refractory period. Absolute refractory. From the firing level up to the 1/3 of the repolarization is the absolute refractory period. Whatever may be the strength of the stimulus, within this period, this nerve cannot be stimulated again. That is called absolute refractory period, during which whatever may be the stimulus strength, it cannot stimulate this nerve or muscle within this period. It is the absolute refractory period. So your basic concept that what is the reason can be reasoned by the absolute refractoriness, because in this condition most of the sodium channels remain in the inactivated state. Suppose you consider that the sodium channel, it is a sodium channel, and due to some changes, some arrangement, it will coming and it is in the inactivated state. Whenever it is activated, the sodium channels are open and they are allowing sodium out. But it is opening and it is quickly in the inactivated state. And when it will gain this our normal state, then there is a closing. There is outer gate is open, inner gate, outer gate is closed, inner gate is open, then normal state, and in that condition it will be activated again. So it is the inactivated state, and when it gains the normal state, then it can conduct another impulse, it can open again. Absolute refractory period, from the beginning of the threshold to the 1/3, roughly 1/3 of the repolarization is called it is the absolute refractory period. And from this point to the beginning of the after depolarization, this part is relative refractory period. Because within this time, some of the voltage gated sodium channels, they are gaining at this part. It is a statistical concept. They are working together, but they are opening at different times, slightly different times, and they are closing at this slightly inactivated, slightly different times. So there are some during these conditions, some sodium channels, they are out of this inactivation. normal state. So they can be stimulated if the stimulus strength is more. It is relative refractory period. And within this period, after this period, after depolarization, and it is sufficient amount of sodium channels are opened, so that in that case there is some increase in the excitability, because it is close to the firing level. But after that, after the end, up to this end of this, then again its excitability is low. It is to some extent relatively refractory. As this after depolarization phase is very slow, so some texts they consider from this point relative refractory period, they do not ignore it, they ignored it. The absolute refractory period, and up to this it is called relative refractory period. From the beginning of this, from the firing point, there is this spike potential, and that is a 1/3 of the repolarization, and it is the absolute refractory period. No stimulus, whatever may be the strength, will excite it again. And after that, from this point to the beginning of the after depolarization, it is the relative refractory period. In some reliable text they declare, because within this period some sodium channels are regaining this normal position, and they are able to conduct sodium ions. Whenever the stimulus strength is more depolarizing. But from that point, throughout the after depolarization, suppose it is 40 milliseconds or so, it is a prolonged period, and it is away from this firing level. So in this case, excitability is also reduced. So it can be concluded into, it may be included into the period. So some good texts, they become simplified, and after depolarization is a very small part, so that they consider after the absolute refractory period, then to the end of the after depolarization together, they consider it is the relative refractory period. It is a simplified version, and it is also scientific. But some good texts see that during this phase, there is, it is this, there is this excitability will increase, and during this period there is no excitability, but here there is some increase in excitability. Now again there is lack of, there is the excitability is lower. So initial part of the spike potential from the point of, that is the firing level, up to the 1/3 of the repolarization, it is absolute refractory period. No stimulus, whatever may be the strength, can stimulate it within this period. And absolute refractoriness is due to the inactivated state of the majority of the sodium channels. And from this period to the beginning of after depolarization, some sodium channels are become normal, and they can conduct sodium ions, and with increased stimulus strength or depolarization, it can respond. So it is called relative refractory period. And after this, and when it reaches the baseline or estimate potential, or this period after hyperpolarization, it is also away from this. So more strength is required, stimulus strength is required. So it is also included in the relative refractory period. But if you answer in this way, that the initial from the period to the 1/3 or half of this action potential of the repolarization is absolutely remaining all part, including the active hyperpolarization, is relative refractory period, you are also correct. Because after depolarization is a short process, very short time, 4 milliseconds or less given in the text. Yeah. So this is this concept. After it is the, and in the electronic potential, if it is cat electronic potential, and in this way, this excitability increased, because it is moving towards this firing level. On the other hand, if it is an electronic potential of this magnitude, and in that case it is away from this So there is this stimulus strength, then there is excitability is reduced. Excitability is reduced in an electronic period. This is something about this refractive period. Yeah. And the distribution of these voltage gated sodium channels are different in different part of the cell. In case of a motor neuron, if you consider that is present in the anterior horn cell, at its surface per square micrometer, it is 50 to 75. 50 to 75 in this cell body, and at the initial segment it is to some extent more, so that impulse will start at this point 350 to 500. In the nodes of Ranvier, the per micrometer square it is 2000 to 12,000. Over the myelin it is less than 25 only. Overall there is some rounding, and voltages that develop will be reduced according in every layer. So it is a almost perfect insulator. Yeah. And at the termination, there is the terminal buttons. It is 25 to 75 micrometers. In unmyelinated actions, it is on the average it is 110 per micrometer square, and it is maximum at the nodes of Ranvier and at the initial segment, so that the impulse can generate in this way. And this will be the site of, there is this generation of the action potential. Time is up. So next day we will discuss some part that is remaining, and also to some extent your old osmosis, body fluid, and how this equilibrium is maintained also.